last lecture for chapter 6. We're going to be talking about enzyme regulation. So remember, from the last lecture, we talked about how every chemical reaction in a cell has to be catalyzed by an enzyme. Well, if you look at this figure, that is all of the chemical reactions, or most, in the human body, would it make sense that all of those reactions are taking place at the same time? And the answer is no, because some of those reactions are anabolic reactions. They're building things like DNA and proteins and RNA. Some of those reactions are catabolic, where they're breaking things down. Well, you don't want the cell to break down something you just built if that's not necessary. That would be a waste. So enzymes have to be regulated. So how does that work? Well, there are a variety of strategies that the cell can use to regulate enzymes. And so enzyme inhibition is the idea of basically turning enzymes off or slowing them down. So if we look at panel A over here, we have an enzyme that shows the substrate binding to the active site. That's what happens under normal conditions, no inhibition. Panel B shows competitive inhibition. And in this case, there is a molecule called an inhibitor that when that molecule is present, it can also bind to the active site. It essentially mimics the substrate. So if the inhibitor is bound in the active site, the substrate can't bind. But notice the inhibitor doesn't have the exact same shape as the substrate. It's just similar enough that it can bind to the enzyme and block the active site. So in this situation, the substrate and the enzyme are competing for binding to the active site. They're trying to get to the same place. So in a competition, you can tip the balance of the competition in favor of one side or the other. So if a cell really needs to slow down an enzyme by competitive inhibition, it can increase the concentration of the inhibitor. If it needs to tip the competition in favor of the substrate, it can decrease the concentration of the inhibitor. Okay? Now notice when the inhibitor binds, there's no reaction that takes place. The other important thing to know about enzyme regulation under normal circumstances in cells is that regulation is reversible. So in other words, the inhibitors don't bind to the enzymes and stay on them forever. They're going to bind for a little while, and then they're going to come off, and then they're going to bind again, and then they're going to come off. Well, every time the inhibitor comes off of the enzyme, there's a new competition between the available substrate and the inhibitor. So if the substrate wins the competition, then a reaction is going to be catalyzed. Okay? So it's not permanent. Permanent inhibition is bad for living things. Okay? Permanent inhibition or irreversible inhibition is what a lot of poisons do. Heavy metals can be irreversible inhibitors, where you're essentially poisoning a living thing. You're permanently inhibiting its enzymes so that it can't do the job it needs to do to support life. And that's a bad thing. Okay, so we want our regulation to be reversible. We don't want to permanently disable the enzymes. All right, so competitive inhibition is one possibility, and it's sort of the simplest, least robust form of enzyme inhibition. Another form is non-competitive inhibition. For non-competitive inhibition, we've got the active site, but then we have what's called an allosteric site. And the prefix allo just means other. So there's another binding site on the enzyme. So in this case, the substrate and the inhibitor are not competing for the same binding site. If the inhibitor binds the allosteric site, it is going to cause the enzyme's shape to change. And when the shape changes, the substrate can no longer bind the active site and the enzyme can no longer induce its fit around the substrate to catalyze the reaction. So while the allosteric inhibitor is not physically blocking the substrate from binding, it causes the enzyme shape to change so that there can't be a productive enzyme substrate complex that forms for the reaction to go forward. Now again, the inhibitor, the non-competitive inhibitor, is not permanent binding, so it's going to bind and it's going to come off. When it comes off, the substrate can bind if another inhibitor doesn't bind first and cause the shape of the enzyme to change such that the substrate can't bind. Well, cells don't just want to inhibit enzymes. Sometimes enzymes need to be activated. So allosteric regulation is a broader phrase that includes both activating and inhibiting enzymes. Allosteric regulation, as we said before, means that there's more than one binding site on an enzyme. Okay, so if we look at this top panel, if we look at the top half of the figure, we have an enzyme with 4 subunits, which means it has quaternary structure. And there are active sites. Each enzyme has an active site. And there's also regulatory or allosteric sites where activators can bind. So when an activator binds the allosteric site, it is essentially stabilizing the active form of the enzyme so that the substrates are more likely to bind. But these allosterically regulated enzymes, they automatically or naturally flip back and forth. They oscillate between the active form and the inactive form. And notice how different the shape is in the inactive form compared to the active form. Well, in the inactive form, there are also allosteric sites, but instead of activators binding, inhibitors are going to bind. And so when inhibitors bind to those allosteric sites, the active site is no available available to the substrates, and so they cannot bind. Okay, and notice it says at low concentrations, activators and inhibitors dissociate from the enzyme, and then the enzyme can oscillate back and forth between active and inactive until another regulator binds to it. Okay, so again, these are not permanent on switches or off switches. It's just a way for the cell to help regulate enzyme activity in order to control metabolism. Well, another common mechanism of inhibiting and regulating enzymes is through feedback inhibition. Feedback inhibition takes place when we have metabolic pathways. And if a pathway is just what it sounds, okay, it's, substrate A being converted into substrate or into B, and B is converted into C, and C is converted into D, and D is converted into E. Okay, and so A is our initial substrate. So enzyme, I'm going to abbreviate this NZ. NZ1, enzyme 1 converts A into B. Well, B is the product of enzyme 1, but B is the substrate for enzyme 2. Enzyme 2 converts B into C. C is the product of enzyme 2, but is the substrate of enzyme 3, and so on. Okay, so in our figure over here, we're looking at making a couple of different amino acids. Okay, in this case, we're talking about creating isoleucine, which is one of the amino acids. The initial substrate is another amino acid, threonine. Okay, so threonine is the substrate. It binds in the enzyme's active site. Okay, intermediate A is created. Enzyme 2 converts A into B. Enzyme 3 converts B into C. Enzyme 4 converts C into D. And enzyme 5 converts D into isoleucine. Well, the cell, like I said earlier, needs a way to regulate these pathways, because just because threonine is available doesn't necessarily mean that the cell needs more isoleucine. So if the cell has enough isoleucine, if the concentration of isoleucine is high enough, then it is going to bind to the allosteric site of enzyme 1. Okay, and so that's what this red arrow and now green arrow is showing. Isoleucine binds to the allosteric site. When isoleucine binds to the allosteric site, it shuts down this pathway at enzyme number 1. However, when the cell uses up all of the isoleucine that's available, the isoleucine is going to dissociate or come off of the allosteric site, and now our enzyme is available again. Threonine can bind, and this pathway is reopened. Okay? So why wouldn't it make sense for isoleucine just to feed back and inhibit, say, enzyme 4? Unless the cell has other uses for A, B, and C, it doesn't make any sense to inhibit the pathway far down the pathway. It makes a whole lot more sense to inhibit the pathway at the beginning. Okay? Because there's no point in doing these other 3 or 4 reactions unless the cell has other uses for those intermediates. So the product of the reaction of the pathway, when the cell has enough of that product, it's going to feed back and shut that pathway down through allosteric regulation. Okay, and again, that's reversible. So when the concentration of isoleucine goes down, these isoleucines that are bound as inhibitors are going to come off. The enzyme is now back to its normal state. If threonine is available, threonine can now bind the active site and the pathway is open again. When isoleucine builds up to a high enough concentration, it's going to shut the pathway down again. And so you have this give and take that is sensitive to the concentrations of the amino acids in this case and other molecules in general within the cell. Okay? So that's an important mechanism of inhibition. And feedback inhibition is nice because it's a product of the pathway that acts as the inhibitor. There doesn't need to be some sort of outside molecule to come in and inhibit the pathway. It's just a product of the pathway itself. So it helps keep things a little neater and tidier for the cell. Okay, so I just want to review a couple of things. All right, so remember in most of our conversations about enzymes, we're talking about proteins that are enzymes. Well, where do proteins come from? Proteins come from gene expression, which again, remember, is transcription of genes into mRNA, and then translation of those mRNAs into proteins. Does every cell express every gene? And the answer is no. Okay, we've talked about this before. What this means is that different cells have different collections of active enzymes. Okay, so the liver, for example, is an organ in animals that detoxifies substances. Well, that's the liver's job. That's not the heart's job. So the liver is going to express lots of enzymes that do detoxification reactions. The heart doesn't need those enzymes. So the heart is not going to express the genes that result in those enzymes being built. So the heart's not going to have those enzymes be active, but the liver will. Okay, so it's all about the jobs that different cells have to do. Okay, now are there some enzymes that pretty much all cells have? Absolutely. But when it comes to the specialized jobs of different cell types, they're going to have different enzymes to do those jobs. Okay, so that big metabolism slide that I showed you in the beginning, not all of those reactions are happening in every cell. Okay, some of those reactions are specific to different cell types, and therefore those cells are only going to express the genes for the enzymes that they need. Okay, the other thing that I think it's important to remember is that enzymes catalyze reactions, but the product of those reactions aren't necessarily proteins, right? The product of enzymatic reactions can be any kind of molecule. When you watch the biology of skin color, you learned about how different enzymes work in the pathway that creates melanin. And some people, based on the alleles that they have, will produce the pheomelanin or the lighter melanin, and some people will produce the eumelanin or the darker melanin. Okay, well, melanin is a product of a metabolic pathway that is catalyzed by enzymes, but melanin itself is a pigment. Okay, so when we have genetic disorders, say albinism, albinism is a lack of melanin. Well, that results because an individual has a mutation in a gene that codes for one of the enzymes in the pathway that creates melanin. If the enzyme is missing, the reaction can't happen, and therefore the organism doesn't create that final product. In the case of albinism, that would be melanin. Okay, so it's not that there's a gene, one gene for melanin. Melanin is not a gene product. The enzymes that create melanin through chemical reactions are the gene products. Melanin is the final product of that biochemical pathway. Okay, I hope that makes sense. We will have some opportunities for you to ask questions about these lectures for Chapter 6. I'll be posting that information soon, and we can meet virtually, and you can ask